Hey, good morning, everybody, and happy Friday. So today we're going to talk about RNA targeted pharmacology. And the last time we talked about pharmacology that targets DNA, and RNA indeed is very similar to DNA. It's a nucleic acid. But what you're going to find is that the pharmacology, when you target RNA as opposed to DNA, is actually quite different. So this actually represents an area of kind of intense innovation that's going on right now in pharmacology. And the whole idea is that you can kind of hack the system. So you all know that genes are made of DNA. They make a copy in the form of messenger RNA, and then that messenger RNA makes protein, right? So the last 100 years of pharmacology has mostly been about targeting proteins. And every single protein is different. So the process of drug development is totally specialized for the particular protein that you're working on. But this technology promises to really expedite things because all proteins have messenger RNAs, right? And one messenger RNA is kind of similar to the next messenger RNA. They only differ by sequence. So this is a technology that targets a particular sequence of an mRNA, and if you target the mRNA, then you can, for instance, inhibit making the protein at all. So instead of inhibiting a receptor, for instance, you could inhibit the messenger RNA and get less receptor made. And the neat thing is that all you really need is knowledge of the sequence of the gene in the messenger RNA to do this stuff. And you don't have to kind of reinvent the wheel every time you want to make a pharmacological product. Because how you target one messenger RNA with these approaches is very similar to how you would target the next and the next. And so it's a really interesting area, and I think you'll see much more of this going forward. So let's get into it. As I just mentioned, genes are made of DNA, but a gene is nothing but information. So in order for that gene to be expressed, to make a protein, the first thing that has to happen is transcription occurs, and a messenger RNA molecule is made that has a sequence that is specified by the gene, and then that messenger RNA has to be translated in the ribosome, and you finally make a protein. So DNA making RNA is the process of transcription, and RNA making protein is the process of translation. Most pharmacology is focused on directly targeting proteins, but this new approach is targeting the RNA that encodes the proteins. Okay. So throughout this lecture, this slide here, this is just so you can go back and study it. There are a lot of abbreviations. So if you get lost, go back to this slide, but I'll try to define them on first use when I'm speaking here this morning. mRNA is messenger RNA. miRNA is microRNA. And all of this technology basically mimics microRNAs. So I'm going to tell you about what microRNAs are and how they work. Some of you may have seen that, or maybe just a bit familiar with it, but microRNAs are very interesting. Almost every single messenger RNA that encodes every protein in our bodies is subject to microRNA regulation. RNAi is RNA interference, and it can be natural, but it can also be mediated by microRNAs, but it can also be induced pharmacologically. siRNAs are small interfering RNAs. ASOs are antisense oligonucleotides. RISC is the RISC complex. This is a protein RNA complex that works on messenger RNAs. And within the messenger RNA, be familiar with these three segments: the 5' UTR, the ORF, and the 3' UTR. So this is the untranslated region near the 5' end of a messenger RNA. The ORF is the open reading frame. That's the part that specifies protein. And the 3' UTR is the untranslated region toward the end of the messenger RNA. But this will all shake out as we go ahead and go through this. So what I'm going to do first is talk about mechanisms that occur between transcription and translation. So there's actually regulation that goes on in all of our cells after messenger RNA is made. And so there are multiple levels of regulation that occur at the level of RNA. Of course, the first one is just transcription itself. That's making a messenger RNA from a DNA template. But the second thing that goes on is intron removal. So all human genes, most human genes, I should say, have introns. And these are regions of sequence that are spread out through the ORF and also the UTRs, the untranslated regions. And they get removed by the process of splicing to make a mature mRNA. Interestingly enough, a lot of these introns, you probably were taught that they're basically garbage that you splice out and throw away. It turns out that a lot of introns contain microRNA genes. So these microRNAs, some of which are derived from the introns of pre-messenger RNAs, these are short non-coding RNAs. They don't code for protein themselves, but they can actually regulate the levels of translation of a messenger RNA, so how much protein is made in the ribosome, or they can even regulate the stability of the mRNA. They can destroy the mRNA, reduce its levels. And so that's an interesting thing, and we'll go into that because that's the basis of the pharmacology that's been developed, mimicking microRNA function. And of course, there's also regulation of the stability of messenger RNAs that occurs that has nothing to do with microRNAs. They all have intrinsic half-lives. There are other processes, like the 3' UTR is kind of protective of the stability of the messenger RNA. Polyadenylation affects the stability of the messenger RNA. There's another process called nonsense-mediated decay. We won't go into that. But anyway, all this stuff has to do with how long messenger RNAs persist in the cell and how well they can be translated. And of course, that's going to affect how much protein is made. So, just a brief review of transcription. You've all seen this in basic biology. But what happens is, let's say this is a gene. The sequence is here, and that's present in DNA, so it's present as base pairs. And one strand codes for protein and the other one does not. But what happens is, in order for the gene to be expressed, the double helix opens up, and then RNA polymerase, an enzyme very similar to DNA polymerase, comes along and makes a copy of that sequence, but now it's using ribonucleotides instead of deoxyribonucleotides. And once you get to the end of the gene, the mRNA is released. And it's a copy of the gene, basically. And then, of course, that goes on and gets translated in the ribosome, and you make a protein. The first copy that is made, though, of a gene is called a pre-messenger RNA, and that pre-mRNA contains introns. And these introns can interrupt the open reading frame, and they do need to be removed to make a mature messenger RNA that then has a completely intact open reading frame, an ORF, that can be translated into protein. And so this is how intron removal happens. Here is an exon. That's part of an ORF that would encode genes using the genetic code. And here's the intron, and here's the next exon. Like this might be exon 4, and this is exon 5. And there's an intervening sequence, an intron between them. In a multi-protein complex called the spliceosome, the first thing that happens is the 5' end of the intron is cleaved, and then the 5' tail of that intron gets attached internally to an adenine residue, and then the 3' end gets cleaved, and the intron gets released as a lariat structure, because it looks like a cowboy lariat, and the 3' end of this exon gets joined to the 5' end of this exon, and now that intron is removed, and this is now a functional mRNA, basically. So that happens to all of our genes and pre-messenger RNAs. Turns out that some of these microRNAs are actually encoded in these introns. So they're not just garbage. They contain some valuable regulatory small little RNA molecules that regulate messenger RNAs. And that's all I'll say about that. But let's get into these microRNAs now. What are they and how do they work? And so this is the basis of the pharmacology that we're going to talk about. But in order to understand the pharmacology, you need to understand microRNAs. And here's how it goes. So microRNAs perform a function called RNA interference, which is to interfere with the stability and the translation of messenger RNAs. And what they do is something called silencing. So silencing is basically anything that prevents a protein from being made by a messenger RNA. So just because you have a messenger RNA that's made doesn't mean that you're going to make protein. And one of the reasons that that happens are these microRNAs. So there are two basic ways that the activity, the coding activity of a messenger RNA can be inhibited. And one is you can prevent that mRNA from being translated into protein. So even though the messenger RNA is there, some of these microRNAs inhibit the production of protein from the messenger RNA. They don't destroy the messenger RNA, they just inhibit its translation into protein. And the second way that silencing can occur is that through RNA mechanisms, that mRNA can be destroyed. So if you don't have a messenger RNA present, you can't make a protein, right? And so those are the two methods. On the one hand, inhibiting translation without destroying the messenger RNA, and then the second way is to just destroy the messenger RNA. And this happens all the time. And so these microRNAs can act by either of those two mechanisms. But each particular microRNA will use one or the other mechanism, okay? So here is how they work. And this is actually a very important slide to understand. MicroRNAs are tiny little RNAs, and they don't code for protein. They're usually about 20 or 21 bases long, so they're tiny. But microRNAs, many of them have their own genes. Some of them are processed out of introns, kind of half and half. But the point is, somehow or another, here's an example of a microRNA having its own gene. So RNA polymerase comes and makes a primary microRNA, which is called a pri-microRNA, and it looks like this. So RNA, even though it's single-stranded, can form internal base pairs. And this is looking at a primary microRNA. Here's the 5' end here on the left, 3' on the right. And internally, even though it's single-stranded, you can get base pairing, okay? And so this stem is going 5' to 3' from bottom to top, and then 5' to 3' from top to bottom. And so there's internal base pairing. And you get things like loops and little bulges where base pairing isn't present. So the first thing that happens in the nucleus is there is a protein called drosha, which comes along and cleaves off that hairpin stem loop structure. And now we would call that a pre-microRNA, okay? And this happens in the nucleus because that's where pre-messenger RNAs are made. Transcription happens in the nucleus. And that pre-microRNA is then exported to the cytoplasm, and it encounters another enzyme called dicer. And what dicer does is it is an endonuclease that cleaves that little loop, leaving only the stem present. Now this particular microRNA happens to have a little bulge in it. That means there's a little region that's not base paired. So you have a little region of double-stranded RNA now, okay? It's then acted upon by a helicase, and what a helicase does is it separates it into single strands, and then one of those strands can associate with the RISC complex, and RISC stands for RNA-induced silencing complex. And one of the proteins in the RISC complex is called argonaute, AGO. And this RISC complex then does something very interesting. It scans messenger RNAs, and it finds one that has complementarity to the microRNA. So it's kind of like a matchmaker. And it will help the microRNA base pair and bind to a targeted mRNA. And this is based on base pairing, Watson-Crick base pairing between the microRNA and the messenger RNA. So it sticks to the messenger RNA by base pairing. Okay? And two interesting things can happen. Most of these microRNAs are about 21 bases long. If it's a variety that has absolute complete complementarity, all 21 bases exactly match the target mRNA sequence, that causes nucleolytic cleavage. That means that messenger RNA gets cut in half at the site where the microRNA binds. And a messenger RNA that's cut in half cannot make protein. And furthermore, that cutting actually destabilizes the messenger RNA because most messenger RNAs have some protective areas at the 5' end, a cap at the 5' end, a poly A tail at 3' end that protects it from degradation. When you clip it in the middle like this, it gets degraded to mononucleotides and destroyed completely. So exact matches of a microRNA and an mRNA result in the complete destruction of the messenger RNA. But interestingly enough, if there is a mismatch, like one base doesn't match or two, then that microRNA can still find its target, but something different happens. In this case, the messenger RNA is not destroyed, but rather what happens is it inhibits translation. So this messenger RNA with these microRNAs stuck to it cannot make protein, okay? And this is a form of regulation that goes on in all of our cells. I would say that, this makes it look like it's an all or nothing thing, right? But there might be some mRNAs that don't have a microRNA attached, and those can go on and make proteins, whereas the ones that do have a microRNA attached will not. So if, the microRNA is expressed at a certain level, you might go from like 100% protein production down to 20% protein production, because maybe 80% of the mRNAs have a microRNA attached. So how much microRNA is there is also critical. The other thing I would say is that microRNA expression is exquisitely tissue specific. So you might have mRNAs that are expressed everywhere in the body, in all our organs, but in one particular organ, there's a microRNA that's expressed, and it targets that messenger RNA, like only in the liver, for instance. And so that protein is not made in the liver, even though the gene is being transcribed. And there are all sorts of evolutionary strategies going back, actually hundreds of millions of years that regulate whether genes are expressed in one tissue versus another. And that's actually the basis of multicellular differentiated life. If all of our cells were exactly the same, we have the same genome in all our cells, right? How come we have all these different cell types in our body and all these different organs? It's because gene expression is different from organ to organ. So if we didn't have stuff like this going on, we would all be like single cell amoebas or something like that. Life would be really boring. But anyway, that's why this stuff exists. But humans have figured this out, and we basically hacked the system for pharmacology. This is another slide I'll show you. It's just to reiterate this important concept. Here's Drosha and Dicer working to go from a primary microRNA all the way down to a ready microRNA. But if that complementarity is complete, you get RNA cleavage and destruction of the messenger RNA. If it is imperfect complementarity, then you get this other mechanism, which is translational repression. So I really want you to know those two differences and why they work. Okay, so that's the basic biology. Any questions so far? Okay. So if you look at this in another way, here is a microRNA in the primary microRNA, and through Drosha and Dicer, it ends up becoming a mature microRNA. There's also some preference for where certain microRNAs bind in the messenger RNA. There tends to be a tendency for the ones that cause complete cleavage to be present in the open reading frames, and the ones that cause translational repression to have targets recognition sequences in the 3' UTR. And that's a little bit weird because you think this is the kind here with the imperfect complementarity, and yet they inhibit translation. So why is the target in the 3' UTR? It turns out that there are proteins in the cell that cause interactions between the 5' end of an mRNA and the 3' end. And so ribosomes can kind of go around in circles and continue to crank out protein. And if you disrupt that with a microRNA, it kind of can decrease translation. Okay. But anytime you find a perfect match, no matter where it is, you're going to destroy that messenger RNA. So RNA has a lot of so-called secondary structure. The primary structure is simply the linear sequence, but the secondary structure of RNA consists mostly of stems and loops. So stems are short little base-paired regions internally within a RNA molecule, and loops are loops. You know, here's the loop here at the end. And you could even call these regions over here tails if you wanted to. So here is, for instance, and notice that RNA contains uracil instead of thymine, and so there is such a thing as an AU base pair in RNA, whereas in DNA you get AT base pairs, right? And that happens a lot. Okay. So this is an example of two microRNAs. One of them is called Lin4, the other one is called MUR1, and it shows you where those microRNAs sit in their stem loop structures. And of course, Drosha and Dicer and then helicase activity will liberate the single-stranded microRNA from those precursors. And like I said, some microRNAs have their own genes, and some are processed out of introns. And interestingly enough, some of the ones processed out of introns regulate the messenger RNA that contains the introns. So that's kind of a cool thing. So tissue-specific gene expression, I mentioned that a bit before. mRNAs and microRNAs both can exhibit tissue-specific expression, but tissue-specific expression of mRNAs is controlled by where the promoters are active in one tissue compared to another. Whereas tissue-specific expression of proteins regulated by microRNAs depends on which tissues the microRNAs are expressed from. So microRNA expression can limit or prevent the translation of mRNAs, but the absence of microRNAs can allow the translation of mRNAs into proteins. So a lot of times you find protein being made in tissues where the microRNA is absent. Okay, so if the microRNA is lacking in a tissue, then you're more likely to make that protein from a particular mRNA. Another thing of relevance for pharmacology is that since these microRNAs typically are expressed in a very tissue-specific way, or an organ-specific way, that means you can get great specificity of drugs that target microRNAs, because if you could target a microRNA, the target is only going to be present in certain cells. Okay? So that can help things sometimes with pharmacology. Okay. So let's get into some of these approaches. The first one we're going to talk about are siRNA. And siRNA stands for small interfering RNA. So what is an siRNA? An siRNA is basically a human-made synthetic microRNA, very similar to it. So we know how microRNAs work, and they're just little sequences of RNA, we can synthesize those in the lab or the pharmaceutical company, and if we can deliver those to cells, then they're going to act like a microRNA. So we can intentionally make synthetic microRNAs, but we call them siRNAs. And what siRNAs are, they're short double-stranded RNAs, but they dissociate into single strands once they get into the cell, and they act like microRNAs. They're synthetic microRNAs, basically. And these siRNAs first got used a lot in labs to knock down particular mRNAs. So it's a great tool because you used to have to go and delete the gene, which could be very difficult in some cases. But after siRNA was invented, instead of going in and doing genetics and deleting a gene, you could just put some siRNA into the cells and it will go along, and usually they're made to be perfect matches. So they do the RNA cleavage thing and destroy specific mRNAs, okay? So they're designed to have a perfect match. And now there are some FDA-approved siRNA drugs, and there are many more in the pipeline. So here's what an siRNA looks like. It's synthetically made double-stranded RNA. They tend to be 20 to 25 bases long, and they have little 3' overhanging tails. But what happens is you have what's called a guide strand, and that's the strand that's actually going to bind to the target mRNA, and a passenger strand that doesn't do much of anything except help you deliver it. But that has to be separated into single strands in order for it to work. And they tend to be 20 to 25 bases in length. There's a magic number in human genetics, and that magic number is 17. So we have 4 possible bases, right? So if you have 1 base, there are 4 possibilities. The possibility of a 2-base sequence is 4 times 4, 16. 3 bases is 64. It turns out that if you have a sequence of 17 bases, usually that's the borderline at which a sequence becomes unique for one and only one gene, because we have 3 megabases of DNA. So if the sequence is longer than 17 bases, it's likely to target one and only one gene. And that's why siRNAs are a little bit longer than that, 20 or 25, something like that. So once you get to that length, it's going to exactly match one and only one target, and you'll have really exquisite specificity, okay? The backbones of siRNAs as drugs have been subject to a lot of interest. Dr. Bahal in PharmSci is very interested in this topic, for instance. And what has happened is that people have found ways to modify the backbone of siRNAs with different chemical derivatives. And what that oftentimes does is increase the stability of the siRNA as a drug. So siRNAs with no backbone modifications have a tendency to be really destroyed quickly in the bloodstream due to nuclease activity. But a lot of these modifications protect from nuclease activity. Things like phosphorothioates, 2'-O methyl groups, 2'-fluoro, 2'-O methoxyethyl groups, MOE, and all of these things stabilize and protect siRNAs because the nucleolytic degradation involves some of these residues, so they get protected. But it doesn't interfere with their ability to base pair, because these are modifications on the backbone, not on the bases. And they just make these into more usable drugs. Another thing that happens a lot is that siRNAs typically, one approach is to place them in lipid nanoparticles, and this protects the siRNA drug payload inside and makes it such that these lipid nanoparticles can freely float around in the blood and reach target cells, and they can deliver them to inside of cells. So it's a delivery mechanism. Another interesting thing that's happened is there's a modification that's put onto the end of specific siRNAs, and it's called GalNAc, which is N-acylgalactosamine. And the interesting thing is there's a receptor for GalNAc on liver cells. And so this binds to liver cells, and then the receptor gets internalized, and the siRNA goes for a ride and ends up on the inside of the cell. So that's a tissue-specific targeting mechanism. And people are looking for other similar things for other cell types, but liver is ahead of anything else right now. So those are all, these up here at the top, these are all stability, structural modifications, and these are delivery-based approaches down below. Okay. And so there are a whole bunch of them. This is just a list of different drugs you can see that have all been approved recently. Department of Pharmaceutical Sciences has a lot of research in this area going on, and you can see the names of them and the disease that they target. They target particular mRNAs, and no one is more important than the other. You don't have to go and memorize every single one of these, but I just want to show you that a lot of these have been approved, and there are many in the pipeline right now. And they're really different than protein targeting drugs. Okay, so the next class, so that's siRNAs. And remember, they're just basically synthetic microRNAs. The next thing I want to talk about are antisense oligonucleotides. So siRNAs are actually made out of RNA, okay? They're made out of RNA. Antisense oligonucleotides, also called ASOs, these are made out of DNA, okay? But they work kind of like microRNAs, even though they're made out of DNA. Okay. So here is how they work, okay? So, and they can work by 3 different mechanisms. So an antisense oligonucleotide is a single-stranded DNA, also something greater than 17 bases long, because you want it to be specific. Usually they're around 20 or 25. And since it's single-stranded DNA, it can actually bind to RNA. So you get an RNA-DNA heteroduplex, base pairing between a single-stranded DNA and single-stranded RNA. That's called a heteroduplex because it's made of 2 things. DNA would be homoduplex. And so one thing that can happen is in the nucleus, it can do things like you could bind it near the promoter and prevent that gene from being transcribed. But a more common thing that's done with this is to impact that RNA splicing that I talked about before, intron removal. You can block a splice site, and then the intron can't be removed, and then that could impact the stability. Another really interesting approach there has been used for genetic disorders, where you know where a mutation is in the patient, and you know it's in a particular exon. If you block splicing of that particular exon-intron junction, you can actually skip an exon and then splice into the next one. So it causes exon deletion, but still makes a functional mRNA. And so you can produce a messenger RNA that doesn't contain the mutation that causes a disease. It splices around it, kind of. And that is basically another approach. So approach 2 is cytoplasmic steric inhibition. And so you have a messenger RNA that's already made, it's in the cytoplasm. Antisense oligonucleotides can work either in the nucleus or the cytoplasm, depending on which mechanism you want to use. But in this case, if this ASO binds to its target mRNA, it can do important things like block translation. So you remember during translation, all translation starts with adding a methionine to a growing protein. Most proteins start with methionine. They're decoding the codon AUG. If you stick an ASO at that region, translation can't get started, and you can prevent protein production. And then the other mechanism, and actually pharmaceuticals haven't really been developed on that approach yet, but there's interest in that. The third one is something called RNase H mediated cleavage. So if you stick a DNA ASO to an RNA, a messenger RNA, you get this DNA RNA heteroduplex. It turns out in the cytoplasm that gets recognized by an endogenous protein called RNase H. And RNase H actually recognizes that DNA RNA hybrid region, and it cleaves the RNA at that site. Okay? And that is a way that many of the approved ASOs work. So ASOs are on the market right now using approach 1 and approach 3. There's some development and interest in number 2, but none of those have made it through the FDA yet. Okay. All right. So kind of a bit of a looking at this just another way. This concept of DNA RNA heteroduplexes is very important. If you have an mRNA in the cytoplasm and you have an ASO bound to it with complete base pairing, RNase H will come along and cleave that messenger RNA, and then it gets destroyed. Okay? The other thing that happens a lot is in the nucleus. If you can stick that ASO, RNase H tends to not be in the nucleus as much, but if you can bind an ASO to a splice site, you can modulate splicing. And sometimes, in the spliceosome, it encounters one of these heteroduplexes, and it says, forget removing this intron, let's just splice out, let's just attach, like instead of going exon 1, 2, 3, you'll attach exon 1 to exon 3, and it skips exon 2. If the mutation is in exon 2, then you don't make a mutant protein. You make a slightly altered protein, but at least it doesn't contain a disease-causing mutation. If indeed, I also want to mention that siRNAs and some microRNAs do use this RISC complex, and that shows what happens with RNA interference. So RNA interference is using this it's more biological, it's using the RISC complex. ASOs are working by these mechanisms we just talked about. So a little subtle distinction there. But they all depend on exact base pairing. So here are some new ASO drugs. You can see they're used for various things, like fomaversine targets cytomegalovirus, CMV replication. Mipomersin targets LDL, low-density lipoprotein receptor. Here's a muscular dystrophy drug, another one. These ones cause exon skipping. I think that's another exon skipping one. And so here's inotersen. It works on a particular genetic disease called hereditary amyloidosis. These have been great drugs for helping to alleviate human genetic disorders. Because the way they work is through genetics, right? They're based on sequence. So pretty cool. Here's an example, using mipomersin as an example, just to show you how it works biologically. Okay, so here is a lipoprotein particle. This is the particle that carries cholesterol around in your body. It contains apoproteins and phospholipids on the surface, and then fats and cholesterol are in the middle of it. And so that helps to solubilize and allows the transport of fats and cholesterol, because they would be completely insoluble if they weren't encased in a lipoprotein particle. So there are many lipoprotein particles, but one of them is called LDL, low-density lipoprotein. That's bad cholesterol. That is LDL. If you have LDL is just packed full of cholesterol. So if you have high LDL, you're at severe risk for heart disease. But these LDL particles have specialized proteins called apoproteins embedded in their surface. And for LDL, it's ApoB100 is the name of it. So mipomersen is an ASO that targets the mRNA encoding ApoB100. So basically, in the presence of mipomersen, the body doesn't make as much LDL because the critical lipoprotein component is not being made. And that's the idea there. I just wanted to make it a little more biological. So, you know, if you block a receptor with a drug, you're going to get biological consequences. If you block the production of a protein with a drug, you're going to get biological consequences. And think about it. Is it really that different to inhibit a receptor with an antagonist for the whole body as compared to preventing that receptor from being present by targeting it with this nucleic acid technology? The receptor doesn't work either way, right? In one case, it's blocked with an antagonist. In the other case, you're destroying the receptor itself or reducing its levels, because this is all tunable. You can reduce the amount of receptor by dosing the drug. And so it's a pretty cool approach. And look for more of this, I think, as time goes on. You know, 10 years from now, there will probably be 10 times as many ASO and microRNA-based drugs around. Okay, the very last thing I want to mention, this is a little bit of a holdover from the pandemic, but mRNA vaccines. How do they work? So here is our friend SARS-CoV-2, the COVID-19 virus. And what you can see is that this is what causes COVID. It's an envelope virus. It has a membrane exterior, but included in the membrane are these so-called spike proteins, and they're glycoproteins. They're modified with sugars. And these spike proteins, and then there's the genome in here, all sorts of other stuff. But the important thing is that spike protein is what binds to receptors on cells and allows entry of the virus into cells, okay? So one of the, remember that because I guess in classic vaccines you would use a bit of the spike protein made synthetically, and that would be a great vaccine because you start making antibodies against spike protein. And actually SpikeVax is like that right now. It's a protein-based vaccine. But the mRNA vaccines require some knowledge of the sequences, the RNA sequences present. And so this is the genome of COVID virus. And you can see that it's a big, long RNA. I think it's like over 20 kilobases, something like that. And it contains a lot of genes that specify different things. But one of these genes actually specifies the spike protein, S, right here. Okay? So remember, if you just like had pure spike protein, not attached to a virus, that would be a pretty good vaccine. But the idea with this is that maybe you can encode that with the messenger RNA. I guess it's just a little more COVID-19 stuff. Basically the same thing, it's just showing you what it looks like. But the important thing is the spike protein is actually expressed from this spike gene right here. And that's what's going to be of interest. So just so you know, here's what happens with coronavirus life cycle. The spike protein binds to a receptor on cells in the airway, and then the whole receptor virus complex is endocytosed, and then the interior, the genome of the virus is released, and it associates with ribosomes in your cells, and you start making more copies of the viral genome. And there's a cool but kind of not so cool protein that's encoded called RNA-dependent RNA polymerase. So this makes RNA copies of an RNA genome, and that's a polymerase that's not present in humans. It's encoded by the virus because we're all DNA-based, but this virus is RNA-based. And it starts cranking out more viral protein. You start making proteins that go into the virus, including the spike protein, and then it gets released. But if we had antibodies against the spike protein, you could have immunity. So if you're immunized, you get infected, or you're exposed to COVID-19, and the viral particles get neutralized by interacting with antibodies against the spike protein. And so that's how mRNA vaccines came about. And the idea here is that instead of actually using the spike protein itself as a vaccine, you make a synthetic mRNA that encodes the spike protein and inject that mRNA, and then that mRNA gets translated by ribosomes, and it's an mRNA that encodes the spike protein, and your cells start to produce spike protein, and then they're released, and then you get an immune response. So it's just an alternate way to deliver an antigen. An antigen is anything that elicits an immune response. And in the case of mRNA vaccines, you deliver an mRNA that encodes the spike protein, and then your cells start to crank out spike protein, and you get a robust immune response typically. And so there are some interesting implications for this. One thing I would say is that this approach, there's been a lot of controversy, quite a bit of misinformation about it presented. But pandemics can emerge very quickly, and we don't know quite where it's really difficult to anticipate the next pandemic. One of the problems is that in animal species throughout the world, there are a lot of viruses present in animals. And in the developed world, there is a lot more contact between humans and various and sundry animals that we probably wouldn't have had contact with originally. But what usually happens is a virus is present in a reservoir of species, and it spills over into humans for the very first time, and you get a pandemic. So the old method of dealing with pandemics is multifaceted. One is to try to contain the pandemic. You get members, CDC and staff members and all sorts of stuff, and you try to prevent the spread and quarantine and isolate people. But if it gets out of that containment, then you can have worldwide infection. And 10 years ago, the only option then is to really study the virus, figure out what the surface proteins are in the virus, start to clone the genes for the surface proteins, express them in E. coli, make the synthetic protein, put that synthetic protein into, and use that as basically an immunogen. You have to mass produce that protein somehow, and that can be difficult. Oftentimes, it's like in flu, it's done in eggs. Eggs are used to make flu vaccine. And so it's sort of a long, tedious process. But one thing that's happened now with our modern genetics is that you can very rapidly sequence organisms, probably like in a day or so, and you can figure out what the envelope proteins are. And now instead of trying to come up with a recombinant protein, you can make a vaccine that is a synthetic messenger RNA. And so the timeline of going from identifying the pathogen to making a vaccine is really shortened by this process. Instead of taking the better part of a year, it might take only 2 months. And a lot of people can get infected in 2 months or in 10 months during a pandemic. So this offers some interesting advantages. I want to spend a little bit of time looking forward, since all of this is kind of an emerging area of pharmacology. siRNAs and ASOs target mRNAs, and so far they've been used for specific genetic diseases primarily. But I think there's a lot of interest now in making them more general, and the idea is they might be alternatives to classic protein-based drugs. So once again, a drug that inhibits a receptor, you could do something very similar by inhibiting the production of that receptor itself. And so siRNA drugs might really replace some antagonist classic drugs. What they're not going to do probably is replace agonist drugs, because agonists activate receptors. And there you really need a ligand-target interaction, and you can't really do that. Basically, the nucleic acid pharmacology is only good at destroying things, but it's not really that good at making things. So bear that in mind. Another interesting idea, though, is that like the Pfizer or the BioNTech COVID-19 vaccines, they make antigens. Maybe you could make other mRNAs that make other useful things. So think about like hemophilia. You lack a clotting factor. Wouldn't it be nice to just get an injection of a messenger RNA that produces that clotting factor? That's an interesting thing. So protein replacement therapies are also pretty attractive in that area. All right, and I think that's where we'll leave it. Keep an eye on this whole field, and I hope you have a bit of an understanding. And it's really different than the DNA pharmacology, huh? Which is mostly about inhibiting replication. If you didn't get a chance to come on Wednesday, have a look at the video I posted, okay? Yep. All right, thanks everybody.